Our first speaker is going to be John Sayud. I hope I'm pronouncing that correctly, from Maria Holland's lab here at Notre Dame. And he's going to be speaking on mechanobiology of dura mater growth.
Yeah. Hi, Professor Rader. If we could actually stay on the first slide for a sec. Thank you. Today I'll be talking about the work we're doing in the command lab with our collaborators to characterize the mechanical properties of the dura mater. The dura mater is this tough outer membrane of the brain. The dura itself, as a surrounding membrane, has important implications for brain folding, and it also plays an important role in mediating between the brain and the skull as the brain grows. Our goal is to use computational and experimental tools to characterize the dura mater's mechanical properties and advance our understanding of its role in the brain folding process. This work is important because the dura is critical to understanding the mechanisms of brain growth, and abnormal brain development can in turn be indicative of several neurological disorders. Next slide, please. What we found through our investigation so far is that much of the brain growth of a mouse after week 4 of its life occurs between weeks 4 and 12. This project is ongoing, and the data we've collected here will be incorporated into our understanding of the dura mater in a manner that I'll describe in more detail in a little bit. We selected mice brains to study because their brains continue to fold and develop after they're born, as can be seen from the graphs here. In this case, cerebral volume is used as a proxy for dura mater growth, since the dura mater is attached to the cerebrum. Next slide, please. To generate these graphs, our collaborators took 7 Tesla MRI scans of 6 mice at 4-week intervals, 3 female and 3 male. These scans were then segmented semi-automatically using the FMRIB software library, or FSL. You can see on the slide a female mouse brain at week 16. You'll notice that the skull surrounding the brain has been removed in a jagged manual segmentation process. After this, the brain we sent through FSL software tools to remove all non-brain material when the volume and surface area can be calculated. The reason this can't all be done automatically is that the FSL software toolkit is built for human brains that have different sizes and shapes from mouse brains, so the software needs extra assistance to be accurate. Next slide, please. We can use the continuum mechanics principle known as the finite growth model to summarize the change in volume of a brain over time to be the result of its cellular growth over time and its elastic behaviors or stretching. The results we've developed so far represent the total deformation of the dura mater. One of the next steps in this project then is to perform tissue cutting experiments on mouse brains and to compare the opening angle of those cuts with expected values from the geometry collected in MRI scans and the growth that it goes through. The group also plans to gather information on the mechanical response of small groups of cells in response to stretch. The device on the right showing such a test is from FlexCell International. These data points together will provide the ability to characterize the dura mater under tension. I think that was pretty quick in general, so if we want to do questions, we can, or we can just move on so we make sure we have time.
Yeah, I'm game for that. Does anybody have any questions?
I have a quick one, John. What MRI do you guys use for these studies?
Yeah, so we work with our collaborators at Purdue. I don't know what the name of the lab is that they work in off the top of my head.
But you're using the MRIs that are located on Purdue's campus?
Yes, they're located on Purdue's campus. Yeah, the 17.
Got it.
Yep, they have a nice resource for that there, and that would be Craig, your collaborator that's doing that.
Dr. Gorgon, yeah.
Yep, cool. What do you hope to achieve out of these next steps? What would be the ideal result that you want to
Yeah, so ideally we would be able to determine, like, if, you know, have a graph of some sort where it's like x-axis is the, you know, like the residual stress or like the elastic like stretching that the brain goes through, and then that y-axis would be like the opening angle, and we can determine like how much stretch is going on in the dura mater as it grows, if that makes sense.
And so if we can bring that full circle, then, you know, how is that data ultimately going to tie back into improving quality of life for somebody?
Yeah, so there are some computational models that are being developed right now that are trying to sort of simulate how the brain grows so that you could maybe, you know, tweak this property or that property of this model, and then maybe generate a brain that grows incorrectly, and specifically grows incorrectly in a way that we see in real life with someone who, say, has schizophrenia. And so ideally, if we incorporate the dura mater into those brain folding models, we can improve those computational models and get them to the point where we can use them to kind of predict and understand more about how those neurological diseases come to be.
Great, thank you, John. Does anyone else have questions for John?
Hey, John. Do you think shear forces have an impact on how the brain develops as well?
Yeah, so most of the research that has gone into like these brain folding models broadly show that the differential growth is like the main way that brains cause themselves to fold, and that's the main mechanism of brain folding, and that's why we're interested in that kind of outer layer and those just the meninges of the brain. Yeah, I don't think people consider it too much usually the shear forces, but that would certainly be a great question to look into.
Well, to make sure we stay on time, let's go ahead and go to our next speaker.
Thank you, guys.
And Alicia, that was a nice segue into your talk, since you're the next speaker. And Alicia is going to be speaking on implantable optical biosensor for monitoring breast cancer treatment. She's from Tom O'Sullivan's lab here at Notre Dame, and I'll let her take it from here.
So hi, everybody. What our lab really does is diffuse optics. So what diffuse optics is if you look around you, you can see all this stuff, right? That's because of visible light. And what we do is we take this small section of the entire spectrum of wavelengths, visible light, as well as near-infrared light. And our goal is to be able to use the small section of the wavelengths to non-invasively or minimally invasively measure cancer progression, or even other areas like the brain, for example. Next slide. So what we started doing here is to create a smart breast clip. Looking at the images first, on the right side, on the top, is a representative image of what a breast clip usually looks like. This is usually inserted through a 12-gauge breast biopsy needle, and it's pretty invasive because ultimately it's just a contrast, kind of like a contrast, so the surgeon knows where to dissect. On the bottom is a figure or a cartoon of what we would like our smart breast clip to be. So you see a zoomed in picture of the smart breast clip. There is a light source, there's a light detector, and it's wrapped in a biocompatible epoxy. The goal is to create a wireless version, so there's not wires sticking out of someone's breast tissue. And with this development of the wireless power as well as a communication device, we can more frequently measure the tumor environment as well as gauge whether or not the chemotherapy is working. So the current prototype is sensitive to the changes in hemoglobin concentrations and oxygenation, but it can also be expanded to other molecular targets like lipid or water. So within the breast tissue, normally it's a very fatty tissue, right? But as tumor grows, the tumor pushes away the usual lipids. So that's where the tumor environment changes. The breast clip we have designed so far is also capable of, or will be capable of the response-guided cancer treatment, as well as development for further or newer controlled therapeutic approaches to breast cancer. Next slide, please. So here's how we did it so far. First, we started with simulations. The simulations really were just photon tracing and seeing how does light move through this tumor tissue. What about if we add like the chip components? How does light reflect or move crazily through the tissue as a result of these extra components? The chip functions really like a pulse oximeter. So if you think of your Apple Watch, how it's capable of telling you your percent oxygenation, your resting BPM, is very similar, except just much smaller. And again, we use visible and near infrared light to determine the tissue properties, such as concentration of hemoglobin. So with this chip design, we're using 2 different wavelengths because that gives us a better resolution in terms of the concentrations of these factors. And we also will have 2 light detectors, one close to the light sources and one farther away. the detectors closest to the light source is really just a calibration method of, okay, how much light is actually coming out. And as a light attenuates through the tissue, that's where the far detector comes in, where it has most information in terms of the light that has passed through the tissue. So the graph on the right is the preliminary testing of our currently wired breast clip inside tissue optical mimicking phantoms that really just demonstrate the functionality of our chip. So we're looking at detected voltage as a function of estimated absorption at one of the wavelengths, 680 nanometers. And as absorption increases, we also see the detected voltage decrease, which makes sense because you can think of it as absorption increases, more photons get absorbed, so less end up working its way back to the detector. So this is just a representative, or one of the many graphs. Next slide, please. So for future work, images on the right are from the first slide, so we're familiar with them. So this work is actually in collaboration also with University of Washington, and they are designing the wireless implant part, or making the current wired version wireless. So just to reiterate, this design has implications in diagnostic and treatment of cancer beyond just breast cancer. If you can imagine, for example, cancer anywhere else in the body beyond breast cancer, this is a much less invasive way because you don't have to constantly go through MRIs or CT scans just to see how the cancer is developing. We can kind of pretty much immediately tell you, okay, is the cancer shrinking? Is it growing? Is there more vasculature growing that probably indicates a growth of the cancer? Pretty much immediately with our design, hopefully in the next year. So furthermore, the University of Washington people are helping us design the wireless power and communication system. And we are currently in the process of starting our biocompatibility and functionality study of our chip inside a mirroring model of human breast cancer. And with that, any questions?
So this is Maria Lieberman. So is the idea that this would sort of be permanently implanted, or at some point you'd fish it out, or what's the?
So it will be cut out eventually. So the way right now we're only focusing on breast cancer.
Yeah.
The goal is to aid in the shrinkage of the breast tumors, but at a certain point, once it's small enough and safe enough to be excised, it will be excised together with the tumor. I mean, the tumor, once a tumor is small enough, it will be taken out together with the tumor.
Could you use this for like fertility monitoring? Like looking at the...
Like whether you're ovulating or not?
Yeah. Mm-hmm.
That's actually interesting. I haven't read any papers on this, but anecdotally, I would think yes, because this is capable of measuring increased blood flow or decreased blood flow.
Yeah.
When you're ovulating, there's obviously an increase in blood flow. And I think potentially, yes. I haven't seen an application in this, but I believe it could be used in that area.
We got time for maybe one or two questions since our speakers are very efficient. I have one, Alicia, since I promised you one. There's a growing research of people interested in how different wavelengths of light interact with cells and tissues, right? Perhaps even in a detrimental or maybe in a beneficial way. So you got this, laser in there at some wavelength. Any consideration to, what effect it might have, just that wavelength of light that you're shining on cell behavior?
As far as I know, cells that respond to light usually have something like channel rhodopsin or some type of optogenetic property. The wavelengths that we're looking at have, as far as I know, like historically been proven just to be like, it doesn't hurt you. However, I don't know on like specifically on the cellular level, like whether or not blue light has been for example, I think C. elegans really don't like blue light. They will move away from that. But I don't know, like in terms of humans, whether or not this is a consideration.
Right. And this is blue. I couldn't remember from your wavelength or.
It's red.
Red.
It's red light and near infrared, so slightly past red.
Yeah, that might be worth looking at because there's been some interesting, it's kind of on the fringe of like, you know, pseudoscience to becoming science sort of stuff, but people interested in how red light may actually increase collagen production and other things with cells. So,
Okay, yeah, I'll look into that.
all right, we should probably keep moving here since we're doing really well keeping on time. So let's go on to our third speaker. Sung-Hoon Roh, who is also in the O'Sullivan lab, is going to talk about an optical-based wire-free breast lesion localization for surgical resection. Go ahead, Sung-Hoon.
Yeah, thank you. Can I move on to the next slide? Much better. Yes. So the localization, wireless localization technique, what I'm working on is pretty much similar with the breast clip, which Alicia presented. But the major difference is my purpose is to localize multi-breast lesions. And the way I can do is just, it'll be much clearer when you take a figure at the bottom. So this small box is what we are going to make for localization devices, and we will put into the multiple tumor lesions. And when we generate the RF signal from the outside, like Bluetooth or Wi-Fi, the inside devices will shine a light, something like the light side. So the way we can differentiate each point is it'll have two light sources. First, one is green light and the other one is a red light. They have different wavelengths. So when we use a red one, we can see the far away tumor lesion. And the green light, we can visualize the closest tumor lesions. That is our idea. And so far now, we have verified the wireless communication works, and also we can excite the two light sources at the same time. Next slide. So the way I did it is we want to combine the optics and radio frequency. So we are going to use optics as diagnosis tool, and the radio frequency, we're going to use this for power transfer. And we finish up the proof of concept by designing PCB and put in the LED and successfully turn on the LED. And we also tested how is this efficient or not by using some kind of electronic word like a S11 and S21 value. And I posted in the figures in the poster, but it just didn't know we need to put the that data into also at this slide too. So at the next slide. So our goal is implant that device into the multiple tumor lesions. So what we need to do is make the device can shine a light even far away, like a 5 centimeter. That is what I'm working on. And second one is need to make a small devices to make it implantable. And the last one is we need to test it in the real human tissue. That is what we're working on, and that's all of my presentation.
All right, thank you, Sung-Hoon. Questions from the audience? Sung-Hoon, what in your initial models, what type of a model are you going to use? Like a mouse model of breast cancer, or what type of model are you going to look at?
At the beginning stage, we're going to test our devices into the pendum and also raw chicken breast.
Okay.
Mm-hmm. Not like layered human tissue right now. Just test the pendum.
And really more benchtop testing, and then
exactly.
And then you talked about human testing. Would there be an animal model, or would you go straight from that to, I guess it would really be an ex vivo human mastectomy specimens?
We will go to the ex vivo experiment. One of our collaborators here, Dr. Candice Ludwig, will give us the real human tissue so we can test it in our device into real tissue.
Very good. Other questions?
Maybe just a quick follow-up to that. So where is Dr. Ludwig located?
Is Bloomington Medical
Great.
School.
Great. And so maybe just to know too, the Harper Cancer Research Institute here at Notre Dame has tissue samples, as does the Indiana CTSI. So if you're looking for, tissue samples and other kind of biological samples, those are two great places to go to look for them.
Yeah, pretty sounds good. Yeah. Thank you for letting me know.
And there's some groups that make models of tumors, like there's cell models.
For
Like-
ex vivo or?
Yeah, yes. So like organelle kind of, or
No, from the Lear human tissue.
From
They will-
human tissue, yeah.
Yeah, exactly.
So
Yeah, I have one something.
Hmm.
Why did you choose those two wavelengths?
Or when we are talking about the absorption spectra of the tumor lesions, they have they can scan different chromophores depending on the absorption spectra. So even tumor is really dense, so longer wavelengths and low absorption spectra will have much more penetration depths. So that is why we chose two light sources. We're still working on optimization, which one will be better for penetration depths.
Last chance for any questions. If not, we thank you, Sung Hoon. And we will move on to our last speaker of this session. Maria Lieberman is here representing her own lab, and she's going to talk about a paper analytic device detects controlled substances found in illicit street drugs. Take it away, Maria.
great. Okay, if you could go to the first slide. So street drugs are both a law enforcement problem and a health problem. So in the US last year, there were 80,000 deaths due to drug overdoses. And the law enforcement activities often fall more heavily on certain communities, marginalized communities. And one of the procedures that happens when police interact with a person who's suspected of possessing illicit drugs is that they'll perform presumptive tests, which are usually color tests that are done right at the scene where the person has been stopped. And the presumptive test is what determines whether that person is going to be arrested and enter the justice system. And in Indiana, many of the people who are arrested as a result of a presumptive drug test plead guilty, and the drug samples are never undergo confirmatory analysis. And this can result in cases where people have been convicted. They plead guilty, but they actually didn't have any controlled substance in their possession. And so the problem is that the field drug tests have a lot of false positive results. And these can be used to sort of lean on the person who has been arrested. And there are a lot of reasons why a person might choose to plead guilty instead of maintaining their innocence. So for example, they don't want to stay in jail for months and months, is a pretty common reason. So we've been working on translating a technology that we originally developed to analyze the quality of pharmaceuticals in Africa. And we sort of have taken that as sort of a reverse innovation, that we're taking it into this drug testing situation in the US, and we're trying to use it in this almost a forensic application. So the testing card is shown in the little picture, and you can see a person is spreading a sample on the card. And after that sample is put on the card, the bottom of the card will be placed in water, and then it will activate a bunch of color tests. So there's a library of different color tests on the card, and it produces a sort of a color barcode. And that can be used to identify different controlled substances. And so in this past year, we've been working with samples of street drugs where we have done LCMS analysis, which is the gold standard analysis. And then we can compare how the ID pad works. So the next slide shows how we set that study up. So we started by using pure illicit drugs, which were obtained from a police lab in Michigan. And these have been, we know that these are relatively pure substances. And then we were able to do a dilution study. And because a lot of street drugs are cut with inert ingredients or with other drug substances. So we've been looking at determining what our limit of detection is and the impact of mixtures on our ability to detect particular street drugs. So for the 4 drugs that are shown here, the accuracies are actually greater than 95% for detecting which drug is present. Limits of detection are between 1% and 4% by weight in an inert matrix of lactose. And lactose is a pretty common cutting agent that's used for these drugs. Then we went to Indianapolis and we worked with the Marion County Coroner's Office. So these are the people that are called to the scene of a fatal drug overdose, and they will collect the body for autopsy. In the case of a drug overdose, the autopsy is typically performed. And then they also will pick up if there's anything that they think might be a drug substance at the scene of the death. They collect that and they throw it out because they don't want other people in the residence to use those drugs. They're already associated with a fatal overdose. So we persuaded the coroners to save those samples for us. So the autopsy involves analyzing the drug residues and drug substances that are found in body fluids in the decedent, is the dead person. But we are now able to analyze the drugs that were found at the scene of the crime and residues from drug paraphernalia like syringes or cookers. And so by comparing these residues and the analyses, we were able to see if the ID pad, this paper test card, could accurately identify the drugs. And in 81 out of 83 cases, we got the same result from the ID pad that we got from LCMS. So LCMS can detect trace components, which the ID pad can't. But in the case of a drug overdose, we're usually looking for a major component. And we do have, the accuracy isn't 100%, so we're still working. So for example, fentanyl is a very potent opioid drug. And so we're incorporating an antibody-based test for the fentanyl, so we can detect it at even lower concentrations. You go to the next slide. The next slide shows sort of where we're going with this. So there's a company that is working with us to do commercialization. So we applied for a grant from NSF, which is a Partnership for Innovation Commercialization Program. And so the company is developing a phone app that can read the paper test card results. And they're also doing they just finished an interesting study looking at whether colorblind people could accurately read the test card results. Answer, not very. So that's another motivation for using the phone app. We also have started a collaboration with the Food and Drug Administration to analyze an herbal product called kratom. So kratom contains an opioid called mitragynine. And FDA has been attempting to schedule mitragynine since for about 4 years. And there's been a lot of pushback from people who use kratom as an herbal product for various, ranging from pain relief to getting a buzz. And right now, kratom is illegal in Indiana, but not in most other states. So it's been interesting doing the testing with kratom. And then lastly, we've been applying the ID pad with harm reduction organizations. So these are organizations that work with people who use drugs and provide needle exchange, HIV testing, and safe injection sites and drug checking services. And so we're trying to figure out what's the best way to give people information if they're going to take an illicit drug so that they don't kill themselves when they're taking the illicit drug. So that's the essence of what harm reduction means, is you're not trying to keep them from using the drug because it's very difficult with people who are addicted, but you're trying to keep them alive. And then maybe eventually they can find access to a drug cessation program so that they can get off heroin or whatever they're addicted to. So that's where we are on this project, and happy to answer questions.
Definitely. Well, I was told we have a hard stop at 1. It's 12:59, so we can take a question or two here. So anybody, I'll offer to the audience first, have a question?
Yeah, when you say they cut with lactose, is that the stuff in milk?
Yeah, milk sugar. Yeah.
Oh, what if they're lactose intolerant? Do they just
Well, it's a
evacuate their
small
bowels?
amount of lactose that's used.
Okay.
So I think it probably wouldn't, lactose, most people who are allergic to lactose, it's not like they're allergic to peanut proteins. It's more it's got to do with the gut microflora. But
Okay. Gotcha.
we developed a set of enzyme-based tests specifically to detect lactose and other sugars that are used as cuts in these illicit drugs. But maybe that would be useful to people who are lactose intolerant and want to test food products.
Yeah. Maria, I'll ask you the last question, and that is, at the beginning, you motivated this really from, I think, a valid criminal justice point of view.
Yeah.
And, these partnerships that you describe here are certainly in line with that, but maybe not directly in line with that. Have you had any interests from groups either locally or regionally in kind of implementing this in more of a criminal justice sort of way? And by saying criminal justice, I mean, not only, obviously making sure that, crimes are prosecuted that should be prosecuted, but also making sure that we don't have innocent people pleading guilty to something they didn't do.
Yeah, in St. Joe County, more than 95% of the people who are up on drug charges plead guilty. Mm-hmm. And so if you believe that the police are that good and that accurate at picking who gets arrested, I don't know.
Yeah.
I think we're going to have a lot more ability to work effectively with police once there's a commercial product and and we work out like the regulatory validation of the phone app. So right now there is no regulatory validation of these drug tests, the presumptive drug tests. There's very basic guidance available from Department of Justice on how you validate the analytical metrics of the test. But of course, you can make the analytical metrics can be pushed depending on what samples you give to the test, right? So if you never show the test things that could be a reasonable false positive, then you'll get great metrics.
Yeah.
So
well, that'll be exciting when you do have your app and your product further along to be able to get that in their hands and see the impact that could have. So thank you to all the speakers and attendees. I encourage you now to switch on over to the primary site for the meeting and enjoy the rest of your day.